From MicrobeTV, this is TWiN, This Week in Neuroscience, episode 67, recorded on December 8th, 2025. I'm Vincent Racaniello. You're listening to the podcast about the nervous system. Joining me today from New York, Tim Chung.
Hello. Hi, Vincent. Hi, Vivian.
Hey.
It's great to be back in this ridiculously cold day.
It's like below freezing today.
It is below freezing. It's not fun to bike.
Also joining us from New Orleans, where it's probably not below freezing, Vivian Morrison.
Yeah, no. You know, it's funny. I just realized that we're both from places that have new in them. New York, New Orleans.
That's right.
Different countries.
Happens a lot in America. Having spent bulk of my year in England, it's just England, but put new in front of everything.
New York, of course. Manhattan used to be New Amsterdam, right?
I did not know that.
The settlers came over from Amsterdam, New Amsterdam. Then we have New Jersey, New Mexico. What else do we have?
New England?
New England.
New England.
That's, yeah.
Well, now I can't think of anything.
And then there are many names of cities that are named after British cities too, like Cornwall.
Wait, that's a Cornwall? That's not a city. That's a county.
Is it?
Yeah.
There's like three different Genevas and Athens in the United States.
Oh, there's a Cornwall, New York. Yeah, so that's not a city. It's a. New York, of course, has a lot of the European names because it was the first settled, right? Well, maybe not the first, but it was a lot of activity. But I don't really know my history. I do know my viruses, and I don't know any neuroscience, although I do have a brain and it functions. So,
sometimes, right?
Yeah, sometimes it functions. Sometimes it doesn't. And
but yeah, the way that it functions varies depending on context and like how you've slept and who you're with and how old you are, and just so many different things go into play. But the paper we're going to talk about today, I think if I were to say like what is the kind of the really big interesting question, like really big fundamental question, at least for me, is how does the brain, which is physically like limited in its size, and it has kind of a limited number of tools to work with inside, based on like physics and chemistry and biology. How does it use these limited tools to create a lot of really rich, like a lot of meaning? How can it, take a distinct number of variables and then reconfigure all those variables to convey useful information? Like you're taking in information about your context and who's with you, and then what are you going to do about it? How are you going to respond to the world? Because that's what everything is based on, I think. It's all about survival, right? It's all about like, how do you change your behavior so that you're like more likely to survive and be successful? If you just like really get down to the heart of everything, all about that. And I think that's a cool question.
And context is obviously very important because every other article you read in the newspaper, the guy's like complaining that, oh, my quote is taken out of context. I'm really not a terrible person. Yeah. It's just out of context. So it tells you that context is very important.
Yeah. And but something else that this is making me think of, because as we'll talk about, the paper's going to be using some images that have certain, that people will have certain like feelings about. So we're going to call it valence, emotional valence. Like, do you feel, is this pleasant or is this unpleasant, essentially? So this is just reminding me of something that I saw in graduate school, which is where like we looked at a face and the face was like surprised or scared, and it was looking straight at you. And then there was another picture where it was like the same face, except for the eyes were directed over your shoulder. And it was like, which one is scarier? And it's the one where they're not directing their fear at you. They're directing their fear at something that's behind you and over your shoulder, which is going to be more threatening. So probably because you can't see it coming. Anyway, that was the explanation. But I think when you break your experience down into these like little differences, like where the eyes are pointed, like there's a lot of information about in there that could direct how you behave.
There's some people who don't look you in the eye ever, right?
I'm kind of one of those people.
In fact,
I'm trying.
I was reading something about TSA the other day, and they said whenever you talk to a TSA officer, you should look them right in the eye, because if you don't, they think you're being evasive.
Shady.
And then there's this group of people who look over your head. Okay, I've encountered many scientists, especially if you just meet someone, that typically look over your head. And I was reading an obituary of Jim Watson a couple of weeks ago, and the author said, I met Jim Watson once, and he shook my hand. Then he looked over my head as if to see if there was someone more interesting in the room.
Huh.
That's damning praise. That happens to me a lot because I'm very short.
Oh, really? Having never seen you in person, I have no idea. I always thought you were very
on the
tall.
internet, everyone's tall.
Yeah.
How tall are you?
I have no idea, but short enough I'm in the land of giant in America, coming to America. That's what I'm going to say.
Yeah, I'm pretty short. Maybe you're my height.
All right, so I guess this would be the time for me to not forget the thing that I always forget, which is to give you the title of the paper as well as the authors. So this paper was published in Cell Reports in 2022. And it's entitled Neural Computations Underlying Contextual Processing in Humans. And the authors are Ji Zhang, Ivan Skellen, and Jack J. Lin out of the University of California at Irvine and then at Davis. And their paper, if I were to skip all the way to the discussion and just tell you, like, what are they doing here? It's an example of science just trying to understand what are the biological or even deeper than that, correlates of behaviors that we take for granted. And so they're providing evidence that there's a mechanism to engage different networks in the brain to encode, to understand and like kind of capture contextual information and then apply that to interpreting facial expressions. And as we said before, this is really important for navigating social situations. Like, I mean, think about how many times a day you do this. Like, right? Yeah, I thought this was really cool because I'm kind of on this kick right now where I'm trying to understand like how does the brain encode information in waves and how in like neural activity translated into waves, and then how do these waves interact to, like I said, capture information, assign meaning to it, transfer it from one part of the brain to another, keeping in mind like the timing of events. So a couple of words that I want to bring up. Valence, emotional valence. Like I said, I'm going to call it kind of like the emotional vibe. It's giving vibes of blah, blah, blah. And it's like, what are you perceiving? How does it make you feel? And they're going to show us that our perception of a face, even one that is neutral, how we interpret that, how we perceive that and interpret it is a function of the context in which the face is encountered. Now, the case of this paper is just temporally close to a certain context. And so the sequence of events is a little bit different than what we're living every moment, because they introduce a context, and I'll get into the task that they're using. They introduce a context and then like half a second later, they'll show you a face. And that's a little bit different than they show the neutral face. And that's different than seeing a neutral face at the same time that you're taking in a context, which they actually say is, that's one of the limitations, is it's not like totally real, like a real life scenario, so to speak. And it's also at a very short time scale versus like a much longer one where like you were in a context 5 minutes ago that gave you some emotional vibe, and then you're presented with a neutral face. Like how much contextual modulation is there going to be when you're perceiving and interpreting this neutral face? So that pretty much leads me to the task this is in humans, by the way. And it's a small number of people. It's 8 individuals. They all have epilepsy. So 5 males, 3 females. And they're taking, so like in epilepsy, at least in these patients, there's an epileptic focus. So there's usually like a specific part of the brain where this, unregulated neuronal activity starts and kind of spreads and leads to seizures. So luckily, if you have that, then they can do surgery to remove it. And so I think, there's not many opportunities to like stick electrodes in people's brains and have them do tasks because you like open their skull. So in situations like this, there's probably scientists that are just like waiting to be like, do you have patients coming in to have an epileptic focus removed? And so they take advantage of that. And for these patients, they'd have the focuses on one side. And so the electrodes that the researchers put into the brain are done on the other side of the brain. But they can't rule out the possibility that the fact that there's an epileptic focus influences these individuals' performance on the task. So what is the task? It has several different stages. There's a fixation baseline, which is where the individual is asked to just fixate on a screen. That's for half of a second. Then they present the context image. And these images are taken from a set of images called the International Affective Picture Set. And this IAPS, they already have, they've like crowdsourced ratings, valence ratings, so pleasantness or unpleasantness. So there's already a pre-assigned value. So, and I think, let's see, trying to see, can't remember if it's like 9 is the worst. Let's see, here, 1, there's the scores between 1 to 9, unpleasant to pleasant. So they also mentioned that in a perfect world, they would select pictures and then show them to the subjects of this experiment beforehand and get their valence ratings of these images. But because these people are waiting to have surgery, they don't have a ton of time, so they have to pick and choose what they're going to do, which is also why they're not doing like longer time scales, where instead of this taking 3 seconds or 3+ seconds, it takes 10 minutes or something like that. So that they mentioned that in their limitations section, that it's possible that it's very likely actually that one person's valence rating is going to be a little bit different than like everybody's feelings. So they have that context image, and it is introduced for 1 second, and they have 3 different kinds of images.
And just to clarify, these context image, they in the paper they show some example. Like the positive one is like a nice little puppy dog running in a field of grass. So I think 99% of humans would rate that as positive. And the negative picture that they show is like someone with a face hidden. You can't see the face, but like holding like a knife to his or her own neck. So I think like also most people would say that's a negative image. And then the neutral image for this context is just like a brick wall. And I guess most people would say it's neutral.
Unless,
so like
unless you're a Pink Floyd listener, and then you have
exactly. So it's so those are some of the examples, but I'm guessing like in these context images, none of them show any faces, human faces.
Yeah. That would be a big problem.
Okay, so it's a non-human face image that somehow us humans have assigned valence. Like, is it a nice image or a not nice image that we like? Yeah. Okay.
It's actually interesting also that they chose to use a dog, because that's like, well, I don't know.
I'm guessing that's just one example. They would have many images.
Yeah, I can't...
that span the entire valence kind of.
Because not
space.
Everyone likes dogs, right?
Exactly.
Right.
Like cats don't like dogs.
Yeah, but I also think like, is it a context or is it another item? Like some of the words that they use in the paper are like context item, like context -item association, and the item in this case is the face. But like but then in the images that Tim was mentioning, like there's an organism in there, the dog, and then the other is like it's a human. There's probably some way to have a pleasant and an unpleasant image that is like literally just like context, like the brick wall is, but like shows carnage or celebration or peace or something like that. I
think a good negative image would be SARS-CoV-2 particle.
Just
most
the
people
virus?
would recognize it,
and
that
would only target virologists. Who's gonna?
People would be like, "What is that? Like pinata?"
Right. Pinata.
But anyway, as Tim mentioned, they have there's a positive and negative and a neutral context image, and it's important that they have these 3 different images because they can then kind of they can pair any given face, neutral face, with those 3 different pictures to say like, it's the same face, and when it's applied or when it's immediately preceded by a positive context, the person's rating, valence rating of that face is positive. When the same face follows a negative context, that same face now has a negative valence rating. And then they also take into account, they collect a valence rating of just the face. Like they just show the subject the face and they say, how do you feel about this face? And the swipe left, swipe right, like
rate. They just call it rate the face.
and also they use like a warm color, cold color distinction. Like that's the person either has to choose a warm color for positive valence and then cool color for negative. So, it's not like a sliding, is it? I don't know. I didn't.
I don't know whether it's sliding or not, but it's not just positive or negative. It seems like there's a gradation because the rating spans from between 0 and 1, at least in this picture, in this graph. So yeah.
So it might not be like a
swipe left, swipe right.
Yeah. But I think it's important to say it's not just like the red dot versus the blue dot. There are because they're going to kind of test the strength of this contextual modulation, which you would not be able to do if it was just a binary, like is it nice or is it not nice. So anyway, we've made it like 3 steps through this task. Well, no, we've only made it 2 steps through. The fixation baseline for half a second, the context image presentation for a second. Then there's a maintenance phase, which only lasts half a second. So it's just a blank screen. And then they introduce the neutral face. And there are 35 faces 17 male, 18 female. And like I said, they collect they collected a face-only valence rating from the individuals, the subjects beforehand. And this neutral face also comes from a similar set of faces that are out there for for researchers to use. The NIM-STIM, where is it? Well, look up NIM-STIM if you're interested. NIM-STIM face set, something like that. So that neutral face is shown for only 1 second. And then we pass to the final phase. Or I shouldn't use say phase because we're going to be saying that word to mean a lot of different things. The next step or stage of the task, which is the decision stage, where they rate the valence, and that is self-paced, so they can take as long or as little time as they want. And they recorded all of that. I didn't explicitly see, at least in the main body of the text, and if that was taken into account, or if anything was yielded from that, like if it took somebody longer if it took the subjects longer given a particular image to decide how they felt about the face. That didn't come up in the main body of the article, so I don't know if it might have been in the methods section, just to say there was no large enough variability or change or specific pairings causing the reaction times to be longer or shorter. So I don't think it was it influenced anything. Okay, let me just see. I think that's everything I wanted to mention about the setup. So the whole point in doing this is because we already know that you can see it, the way you interpret a face or the way you interpret an item is going to depend on the context. It's going to be influenced by the context. So this is not a crazy discovery that they can show that they can do this. Really what they have to do is just establish that they can indeed recreate this in a setting with very limited, variability. There's only a couple of things that they have to worry about, a couple of different variables. Because what they want to do is look at what's happening in the brain. How do different brain regions, which have already pretty much been identified in the literature, how do they talk to each other? How do they capture, encode, and transfer information between themselves to then generate a behavior, the valence rating? Of course, they find that they look at the strength of these correlations between, if you see a negative image, are you going to give the face a negative valence rating? Or same thing with a positive context. And they look at the strength of the correlation, and they find that the correlation for negative contexts is stronger than it is for positive contexts, which, going back to my statement about how everything, if you were to break everything down, it all gets back to survival. And the whole, if you hear rustling in the bushes, it's better to assume that it's a lion rather than a zebra, because if it is a lion and you're oh, it's a zebra, you're toast. So better to just err on the side of caution, right? So, I think I'm of course it would be stronger in a negative situation. So yeah, that's like I said, they come up with a quantitative value for the contextual modulation of context on the face valence rating. They found that it wasn't 100% of trials where there was a congruent contextual modulation. So it was actually 87%, which was interesting. And so the trials that were either incongruent or they showed that there was no modulation of the context on the facial valence rating, they were excluded from the analysis. So for the incongruent trials, there was 8 trials per subject. So that's not a lot. And then there were, that's from the incongruent trials. And then the no modulation trials was only 2. So like, the modulation is pretty robust, pretty consistent. It's just like whether or not it's going to be positively correlated, like they go in the same direction, is like, it's not 100%. So yeah, the conclusion of this section is that there's a strong and consistent modulation of the emotional valence that is applied to a face, or to a neutral face, as a function of the context that immediately precedes it in this task.
So just to reiterate, just to summarize, so if a subject just saw a nice picture of a fluffy dog running in a field of grass, they are more likely to rate a completely neutral, kind of like a passport photo, driver's license face, as being a nice positive valence face, just because they saw a nice dog. Whereas if they saw like someone, like a pair of hands behind bars, which is one of the negative context picture, then they see like a completely impassive face, they would rate the face negatively. And apparently we all do this. And I think they don't mention this in the paper, but and the whole point of the task is they show the context first, and then there's a half a second where they take the picture away, and you kind of have to hold it in your memory, and then they show the face. And there's no mention in the task that we do this kind of quote unquote consciously. We don't look at we're not looking at like a picture of a fluffy dog and saying, oh, it's a dog, I'm gonna remember it's a positive thing. We do it apparently subconsciously, and it just influences our lives all the time. And, things like advertisers take advantage of this all the time. That's why for a lot of adverts, you see a lot of like happy things, and then they sell you something at the end. So they're trying to figure out what is, how this previous context can influence the face valence later on. Sorry, just that's repeating some of the points.
That's important. There's something else that you reminded me of that I was going to say. Well, it's gone now, whatever it was. So maybe it's not important.
We'll come back tomorrow.
Yeah. So like they've established that like they can see this phenomenon at work, and they have to do that so that they can use these electrodes stuck into the subject's brains to see what the neurons are doing. What the brain regions are doing. So brain regions that are thought to be able to like that are functionally connected, or that like integrate their functioning in this kind of way. Like the hippocampus and amygdala, those are words we've probably heard before, and they're located in the temporal lobe of the brain, and then the orbitofrontal cortex, which is in the front part. Those have been implicated. And it's thought that between like different regions in the brain, there's this kind of low frequency wave or oscillation that like drives the binding together of higher frequency activity happening in different parts of the brain. So it's kind of like if there's a lot of little rivulets that are entering a kind of slow flowing river. I'm trying to think of different kind of visualizations of this, because it does kind of get a little bit funky when you're like thinking about, okay, well, there's a theta oscillation that's being generated by this region, but at the same time, it's also having gamma activity, and it, I think it's a little bit hard to visualize, but anyway, that there's the low frequency oscillations in the brain, and their synchronization or synchrony seems to be involved in binding and connecting, functionally speaking, different brain regions like the hippocampus, amygdala, and orbitofrontal cortex, OFC.
I have a technical question. So when they put these electrodes in, how deep do they have to go, and how can they measure what's going on in different parts of the brain, hippocampus, amygdala, frontal cortex, and all that?
So I don't know what the depths were, but the hippocampus and the amygdala are not superficial structures.
Right.
Yeah, they have to go a little bit in.
Mm-hmm.
So you think they have to put the electrode in the hippocampus?
Yeah, they did. They have to.
It's pretty invasive, isn't it?
It's deep, yeah. It's invasive. It's not like Elon Musk Neuralink electrodes, which is just on the surface of the brain, like in the cortex. These will have to go a little bit in because that's where they think the epilepsy or focus of origin might be coming from.
So these are very thin electrodes, I presume.
Yeah, very, very thin.
Okay.
And they drive it in kind of like, and but it's quite a well-established technique, like from quite a long time ago. But then I think the difference is they're driving a lot of electrodes in because they're really trying to map the brain.
And they have to do this. I'm trying to think if they do something like this as a regular part of surgery for epileptic patients, because I must be mixing two things, but they do tests so the patient is awake during these procedures, and they'll actually like periodically test to make sure that the person is like able to complete a certain task, just to kind of check on what they're taking out. But that probably doesn't involve actually sticking electrodes in the brain, I don't think. I think that's just they ask the person to do something.
But they might do things like they might temporarily inactivate a brain region. So these electrodes, they don't just record, they can also stimulate. So Vivian's last TWiN talked about stimulating using electrodes. And they can ask the question, well, if I'm zapping, let's say I think this area of the hippocampus is contributing to epilepsy. Do you actually need it? Can we take it out? So the way they can do it, they can like try to vocally stimulate it a little bit, and that would disturb that area of the brain. And then they would ask you to, say a sentence and see if it's affected by it. And if it's not affected by it, then that's a go ahead for them to take it out, I think.
Mm-hmm. Yeah, that's a good point. And that's something that they, either because of time or IRB, or that just wasn't the point of the study. It's probably it's not part of the study's question, is to try to modulate, see if they can disrupt the contextual modulation by throwing off some of the synchronization and coincidence of certain of the theta oscillations and gamma activity that we'll talk about later. So they didn't do anything like that. But like I said before, this is just kind of a paper that's like, we just want to observe in detail, what is going on while this task is happening. It's not their goal to confirm that, to discover and confirm using like a kind of, knockout or inhibition approach, these pathways, if that makes sense. So I think we've talked about brain waves and oscillations before, but the ones that they're going to talk about in this paper are theta oscillations, which are the low frequency waves, and then gamma oscillations, but they're very high they're high frequency, and they call them bursts here. So you'll have to remind me, Tim, what hertz is. It's something over something.
That hertz is just occurrence per second,
right?
How many times something happened per second.
So 4 to 12.
So theta is 4 to 12. So that means, so when you put an electrode into the brain and you're measuring brain waves, also known as oscillation, they call it low frequency oscillation, even though gamma is very high frequency. What you see is basically the voltage, the electric potential difference kind of fluctuates. And that supposedly people think it reflects axons firing, because those obviously involves electric signals going down the axons, but also like dendrites picking up, dendrites depolarizing and stuff like that. And if all the dendrites kind of point in the right way, a similar direction, then these voltage as the dendrites kind of depolarize as they receive signals from axons and stuff like that. As the neurons communicate, let's say, the voltage across the cell membrane change, and you can pick it up using electrode. And they sometimes change very rapidly in these kind of wave shape function, and it's just something that people picked up and found that it is one of those phenomenon that people picked up. And they come in different. Sorry?
Rhythmic fluctuations, like spontaneous.
Yeah, exactly. And they come in different frequencies. Like the slower ones are like theta, and that seems to happen a lot when, I think in rodents, that happens a lot when the mouse and the rat is navigating around the space. And then there's also famously alpha wave, the first one to be discovered, and that's the wave that you have when you are zoned out and daydreaming and stuff like that. And then the high frequency one is the gamma, and those people think it is reflecting actually cells firing very rapidly, and that get picked up as a gamma high frequency wave, and that's like 70 up to 200 hertz, something like that.
Yeah.
Just for comparison, in the U.S., the electricity coming out of the electrical outlets is 60 hertz, right? 60 cycles per second.
So that's low gamma.
Yeah. That's cool to think about when you're like, oh yeah, there's like electricity in can be measured that way.
But that's AC. That's AC, alternating current. Of course, direct current doesn't do that.
No, direct current would just be flat, I guess.
Yes.
But what's interesting is maybe relevant to a podcast. What's interesting is that when you stick an electrode and measure these brain waves, what they find is these different frequencies seem to sometime can act somewhat independently from each other. So like the theta is a component, and overlaying, and Vivian kind of talked about this a bit early on. Theta is a component, and then overlaid on top of it, there can be gamma that comes in every now and then. So so that's kind of similar to, let's say, we're recording to a podcast, and one of us didn't plug in the microphone properly, and there's like this 60 hertz buzz in the background just constantly, and then we're talking on top of the 60 hertz buzz in the background. And if you look at like the recording, the signal of the video or audio recording, you will see a 60 hertz that is constantly ongoing, and then we are just kind of talking over it, and then my voice would have a very different signature, as kind of spectral signature, frequency signature, compared to Vivian's voice, because she maybe is higher because she's a woman.
I don't know about that.
I don't know about Vincent that. Would be lower because he's a guy with a lower voice than I have. And these would come out on top of the 60 hertz, and you could pick those up. And Vincent's audio engineer, tomorrow would be able to actually just remove the 60 hertz, because that's going to stick out very different from our frequency. So you can think of, and this is just all in a audio file signal, right? It's just the same audio wave, but it has all these components in. And it's similarly when you're recording brain waves, all these different theta, gamma, alpha, beta, they can all come and go in their own little components, and they add together and they get picked up. Anyway, so that was a little bit of a background.
Yeah, and
so
actually,
as Vivian said, there's a biological reason for the fluctuation in the brain, right? As different from AC, I guess.
Right. I mean, the fact that they oscillate is similar to the AC current, but they don't fluctuate. I mean, the reason why the AC fluctuates is because the power generator, wherever the power is coming from, is cycling at 60 hertz or whatever. So unless you are literally got your fingers into the mains and electrocuting yourself, you probably wouldn't have a strong 60 hertz component that is ongoing.
Yeah. Kind of related to the background that Tim is talking about, there are a couple of, I think, quotes from the discussion that I think might be helpful to kind of think about why these waves and why these different oscillations are important and how they work together is important. So I'll just read because I'm going to read them because it would take more energy for me to try to put this into my own words, which would end up just being regurgitations of their words. So like I said, their findings provide evidence for a network level mechanism for the brain to capture and encode and carry over information from the outside that we perceive in the outside world to different brain regions, so that we can like use that information to direct our behavior. And based on their findings, which we'll get into, and I'm not going to, for the sake of time and people who don't like listening to numbers, I'm not going to talk a lot about like the numbers or the magnitude or the math stuff. And we're just going to focus on like core concepts and like, what does this finding mean? But they say here, this dynamic of what they're going to tell us they found is consistent with the oscillatory multiplexing framework, where the same structure, brain structure, can use different frequencies simultaneously for communicating with other brain structures, and that phase synchrony, so the way that waves, like how the waves collide, kind of like what's happening in one wave, how does it correlate with the way that this other wave is oscillating, that can decrease or increase. And that would change, like the phase synchrony can decrease and contribute to segregating these two different communication channels, the two different communication channels being represented by different frequency waves. It's a lot of, like I said, kind of like trying to visualize that is a little tough. And there can be this kind of the larger the lower frequency theta wave seems to play can direct like a lot of other waves in the brain to turn on certain like avenues of communication while turning others off, or turning up one and turning another down. This is all happening at really small time scales, sometimes at the same exact time, and that it's this kind of orchestration that is what allows us to collect all this information with our limited brain and then like make sense of it. And they talk in here about how like they're looking at 3 different brain regions, but the directionality of the relation of the communication between like the hippocampus and orbital front and OFC, and the hippocampus and the amygdala and OFC, the directionality is also important. So a lot of the questions that they ask involve looking at 2 electrodes, an electrode pair, and seeing what's happening between 2 brain regions. Okay, so anyway, they found, as would be suggested by the literature, that the strength of the theta waves fluctuated in specific as a function of the different stages in the task. So they measured, let's see, I was going to explain how they measured how they measured this power and the synchrony, but I wonder if that might even be kind of like not that useful.
But before talking about the synchrony, maybe I'll just quickly give the background of the brain region that they recorded from.
Sure.
When the subjects are looking at like context valence, is a happy dog or a threatening knife, and faces. And they recorded from, Vivian talked about this before, I'll just quickly summarize.
Please do.
The hippocampus. And the hippocampus is important because that is the one thing that is really important for context information and also for memory. So remember, there's like this little like 1 second gap where the context puppy dog was taken away and you have to hold it in memory. And if you don't have the hippocampus, like there's a famous patient ICHEM who hippocampus got taken away due to seizure treatment, and he had no memory. He was unable to form new memory. So hippocampus is important. The amygdala is the second brain region that they recorded from, and that's really important for people think for emotional processing. So like a lot of in animal study, they looked at whenever they do fear conditioning, where they pair like a tone with electric shock, the animal would soon be like really freaked out by the tone. And a lot of it involves the amygdala processing the cue. So people think that emotion is involved in amygdala is very important in emotion. And then the last brain region, the third brain region that we looked at is the orbital frontal cortex. And that is important. The most famous patient whose orbital frontal cortex was lost is a guy called Phineas Gage.
We have to have mentioned him before, right?
Yeah, so I'm just going through the greatest hits. But this guy was like maybe born around the 1800s, I think. I'm not 100% sure, but he lost his orbital frontal cortex when like a tempering iron, like this giant iron rod that is involved in rail work or something like railroad building, went through his front of this brain, like through his eye socket up to, and completely destroyed his front bit of his brain. And he managed to survive, but had a huge personality change. So people think the orbital frontal cortex is important for maybe estimating for so-called quote-unquote executive function. So like, how do you judge whether something is important? So maybe for some sort of value is important. So these three brain regions, the authors hypothesize might be important for this task. That's why they looked at the synchrony of these theta waves across these different brain regions. So synchrony literally is just what that means. Is like, do the peaks of the theta waves, do they seem to align to each other consistently? And it's interesting when two brain regions have theta waves that are kind of trough and peaking at relatively the same time, relative to each other, suggests that they might be communicating to each other or are communicated to from a third source commonly.
So as we talked about last time, the hippocampus is also important for management of appetite, right?
Oh, that's the hypothalamus.
Yeah. Oh, right. That's
yeah, different. It's possible that the hippocampus is
But
important.
what was your point? I'm interested to hear what your point was.
The guy who lost his hippocampus, I was wondering how he did with eating.
Oh, no.
But it's not relevant.
Not sure. It's not a major... I don't think there's any mention of weight gain or loss, so I'm guessing that is still okay. Like, he didn't forget that he didn't eat, or he ate... Like, he doesn't eat a fourth lunch, for example, is my guess.
Tim, remind me though. I made a couple leaps here, but how much is the hippocampus involved? Does it have as large a role in memory formation in implicit? Do you know, in implicit memory formation versus explicit? Because I tend to think about the hippocampus as like serving a role in explicit memory.
People think
formation,
it's important for declarative or declarative memory. So like, or episodic memory. So like, what did I do today? Who did I meet? Which did I go home by riding the bike, or did I walk home, or did I take the bus?
You know when it happened.
Right. Whereas, and I think this is work done by, oh no, I've forgotten her name. It'll come back to me soon if my hippocampus works. But the person who assessed patient HM put patient HM on these tasks where these kind of motor tasks where he has to like draw something, but by looking in a mirror, so everything's reversed. And at the beginning, the patient HM did terribly, but he eventually learned how to do it. And they think this is kind of motor learning, or maybe a bit more implicit learning that you can't really verbalize. Those seem to be spared. But I believe there's also the people who question whether they are completely spared, so they might still be involved. And then finally, there's like a third kind of memory, which is, and this is formation of new memory was messed up by losing the hippocampus. Old memory was fine, just like Alzheimer's patients. But there's a third type of memory called semantic memory. Like, these are like, what's the capital of France? So it's not so these are like facts. As opposed to my personal experience. And I don't have no idea whether that was messed up by the hippocampus or not.
Also, I just want to make a point. When you said you couldn't remember something, and this has to do with waves too, and how they synchronize. When you were like, oh, maybe I'll remember it if my hippocampus is working. But if this is a long-term memory that you have, and your hippocampus, you can have long-term, a person can have can recall their long-term memories, even if the hippocampus is compromised, then it would suggest that the hippocampus is not, at least for some memories, I don't know, doesn't play as crucial a role in recall. But recall, I believe, has also to do, because it's tough when you're talking about memory, because it's not like it's stored in like a place. They used to think like, oh, here's where you store this and here's where you store that. And it's not like that.
Neuroscientists have no problem saying this is where the memory is stored. I mean, but no one knows whether they're right or not.
I mean,
like, for example, the finding that the patient exam couldn't make new memories, but old memories were spared, make people think that the hippocampus is really important for the formation of memory, where at the beginning, when you are making a memory, you have to go to the hippocampus, but the storage is like maybe rooted to the cortex. That's where the memory is formed. So there's a hypothesis like that.
And may not require the hippocampus, because just like in, the hippocampus is going to have, it has a bunch of fields of a population of neurons that are going to be creating their own oscillatory patterns. And then, for example, and like the core regions of the cortex do, and like the subcortical structures like the thalamus. And so having like bursts of activity or spindles, which is like a particular waveform occurring in the thalamus, and how that locks in with the phase of the, a wave that's generated in the cortex. It seems like these dynamic activity patterns is really what drives our ability to encode and recall things.
So that's a huge field of study. So, for example, like Yuri Buzhaki, who's a scientist upstairs here at NYU, he's like one of the godfather of like sharp wave ripple and spindles and hippocampal, all these things.
So cool. Maybe we should have him on at some point to talk about these things. So I'm just looking at a note here. When I said I wasn't going to talk about I wasn't going to go into the math stuff, because I can't really explain it very well. I saw a note here of I wrote, ha ha ha, because I want to read this sentence, because I think it just illustrates just like how this information is just not accessible to me. The WPLI, which is the weighted phase lag index, which they use to measure synchrony, quantifies the cross-regional phase synchrony using the imaginary components of the cross spectrum between signals from each electrode pair. I mean, so I looked it up. Imaginary component is a thing. It's actually not imaginary, but it's not like...
It's to do with the square root of 1, like an imaginary number, also known as I. I don't know. Vincent, have you ever heard of imaginary numbers? These are not fake news
numbers. Is that a question or a statement about the imaginary? Because I don't think it has to do with imaginary numbers.
No. Well, what do you mean? Like
it says the imaginary components.
Yeah, it's imaginary numbers. So like the square root of 1 is this thing called I. And it's just, we don't have to get into this, but if you know, just very briefly, I guess we're going to maths now, but I don't know anything about this. So take whatever I say with a massive grain of salt. But you know how normally when you count from, minus 100 to plus 100, you're on a number line. You just go onto like this line. But if you imagine, well, I guess that's why they call it imaginary number. The imaginary number basically expands this number line with a second axis. So like, let's say the number line, the normal number line, or also known as real numbers, is on the x-axis. The imaginary number will be on the y-axis. So you turn the line into a plane. And the whole point of doing this is so that you can spin around in a circle, because you can spin around in a circle in a plane, right? You're literally in a circle. And that turns into a wave function. So your sine wave and your cosine wave would actually reflect you spinning around in a circle. So it allows you to encode oscillations really well. And that's why people use imaginary number for waves. And like it's basically a mathematical convenience that people invented that's super duper useful. And people get frightened away because the y-axis, the new component that people invented, they call it the imaginary number, as if it's not real. But it's just it's just an extra term that people have figured out to. It's like a hack that allows
Cool.
oscillation to be calculated really easily.
That's good to know. Thank you for that, because I was like, what, we're talking about imaginary things now?
But I wouldn't worry about that.
No.
That's just a way for them to
Yeah.
avoid volume conduction. It's for some sort of artifact reduction. So yeah. But
Anyway, I won't drag us through that anymore. But the point is, so they measured the synchrony between the 3 different brain regions that Tim kind of deepened or expanded on. And they found, and they're looking at different stages of the task. So during the context presentation, the network of these 3 regions is dominated by synchrony between the theta, or like theta wave synchrony between, or like within the temporal lobe, so the amygdala and the hippocampus. So context presentation, there's theta wave synchrony happening there. Then during the maintenance phase, which is that half a second between the context presentation and the face display, this profile switches to having enhanced theta synchrony between the amygdala and the OFC and the hippocampus and the OFC. So now we're starting to have the frontotemporal connectivity coming into the picture rather than just oscillations limited in the temporal lobe. And then during the decision stage, there's this the increased theta synchrony between the amygdala and the OFC remains high and dominates the network activity, which makes sense based on what Tim was saying earlier about how we think of the amygdala as being like a hub for emotional processing. And they found, I may have said something about this earlier, but here it is in play. They find that there's an increase in synchrony in certain frequency range, while at the same time there's a reduction in like the frequency bands that are outside or neighboring, which kind of links back to what I said before about how these brainwaves are able to kind of like turn down the gain in certain places and turn up the gain in others, and that in and of it, that combination has a meaning. They go deeper into in looking at the synchrony between these different brain regions and trying to see, well, if there's a pair of electrodes like the amygdala and OFC, which one is like driving the synchrony? Or is it like one-way communication or bidirectional communication? And that also they found is segregated based on the different stages. So in context presentation, they have, like I said, it's temporal lobe structures, and the amygdala and the hippocampus are communicating to each other. During the maintenance phase, we have the hippocampus that's driving activity in the orbitofrontal cortex. During face presentation, it switches to having the amygdala and the OFC like bidirectionally communicating. And then the decision phase is implicating pathways from the OFC to both the amygdala and the hippocampus. So we're starting to break down kind of like which brain regions are generating synchrony, driving it during these different tasks or different stages of the task. Okay. Now, again, just reviewing my notes to see if this is a section of math stuff that we maybe don't want to go deeply into. I don't know. What do you think, Tim? I was going to start talking about the phase amplitude coupling. I'll just go to my conclusion.
Yes.
Which is that, so actually, what was the phase amplitude? It's understanding how like we were talking about different frequencies of oscillations, the theta and gamma oscillations, how if they are coupled in some way, that this drives associations between context and an item, and it takes into account the timing of like the sequence of events. So the conclusion, without walking through everything else, But they found that there was significant phase amplitude coupling in the theta and gamma bands during the maintenance phase. So that's the half a second block between seeing the context and then being presented with the face. And that it was a theta wave in the hippocampus that was being coupled with gamma bursts from the OFC. And that during the face presentation, it switched so that there was a theta wave from the orbital frontal cortex that was locking in with gamma activity in the amygdala.
Yeah, I was trying to think about how to explain something like this.
Mm-hmm.
It's a little bit challenging, but I can give it a shot. So this phase amplitude coupling just means they observed, so during a trial, you have these, context present, fluffy dog presentation and then phase presentation, yada, yada, yada. And during different stages of this trial, what they find is this theta wave, which is slower oscillating. It's kind of like the underlying wave that is ongoing. It seems to so this theta wave is ongoing, but on top of this theta wave, every now and then you see bursts of gamma, and this burst of gamma wave would suddenly switch on but only at certain phases of the theta wave. So like only when the theta wave is at its trough, when it's at the lowest level. So it's kind of, I don't even know how to think about this, but it's almost like, it's hard to explain. But it's almost like you're kind of like almost, the way neuroscientists have to do it is kind of, the way I picture it is, it's almost like trying to decode like a message that might be from an alien, and it's really hard to understand what the alien is trying to communicate, because the message that our brain is communicating with itself is not in English. Like, it's really hard for us to decode. And suddenly you see like this like really low tone. Whenever you see like in space, you pick up like this low tone, and on top of this low tone, you see this high-pitched chirp, but it only chirps at a particular time during the low tone. You think that that's not by accident. That must be important, and that's what these scientists are picking up on. They find these patterns that only happens at certain stages of the trial, and they are analyze. They first of all report that this exists, and then they're trying to analyze what it means.
Yeah, I think this is what I find so cool, and that I'm still developing the language to talk about it, because it's not the field of neuroscience in which I have any training whatsoever. But that it's these movements across space and time that package information and that are like instantaneous, and they have information that we can use. I just find that like just really cool. So their conclusion after looking at this kind of how the gamma and theta waves fit together is that there's theta gamma coupling occurring in the hippocampus orbitofrontal cortex network, and during the maintenance of contextual information until it's this whole kind of like capturing it and holding on to it for the face presentation stage, and that the association of the context in the face is then encapsulated, integrated by theta gamma coupling between the OFC and the amygdala. So there's this kind of transfer of information between pairs, like brain region pairs that are communicating. Okay, so the next part does get pretty complicated, and based on the kind of difficulty that we've had in finding words to describe what this looks like so that people could visualize it, we might be kind of like stumbling through this part. But it kind of has to do with how the relationships of the different waves evolve over time. And they find that the evolution of the relationship between the theta and gamma activity, that the evolution over time of that relationship is also meaningful. So now we've got like the waves in and of themselves, the fact that the waves are from different regions, that those waves maybe line up, that waves of different frequencies pair up in certain places to encode meaning, and then to capture it and carry it over to a different brain region, and then how those relationships evolve over time and space is also meaningful. It's getting back to the idea that like we've got the limits of like our skulls and the amount of matter that can fit in there and the number of connections that can be made, there's a lot. And how do we reconfigure all of these things? And like how many different combinations of these things can we make and consistently use to package information to then lead our behavior? Tim, do you have anything to add to that?
So just briefly summarizing, the last two figures of the paper leans very heavily on this thing called theta precession. Mm-hmm. So people studying the hippocampus borrows heavily from people studying the hippocampus. And I think it is so complicated, it actually warrants its own paper, podcast discussion on what theta precession is, because it's a very interesting idea about how we might be encoding our environment. And as we move, so this is all based on like so-called place cells in the hippocampus, which is our in the Nobel, so those people who study the hippocampus place cell grid cells won the Nobel Prize, and I think the Nobel snippets, they call it the GPS of your brain. And the way this GPS works actually has a lot of intricacy with these theta waves, at least in rodents, less so in bats, as people recently found. But I think it might be a bit tough to explain it in the remainder of this podcast, but it seems like something that is important in aligning how your different place cells might fire appropriately in sequence. Something similar might also be happening for you to encode how the context might carry this, whether it is a happy, fluffy dog versus a negative knife to your throat kind of valence. And they can see kind of this package of information going from one brain area to the next. I think is maybe the most broad way of summarizing it.
Yeah. And I think this is supported by the literature. They actually say that the gamma bursts, the literature suggests that gamma burst activity actually represents items in a sequence. And then that their alignment with either the earlier or late phase of the theta wave kind of like pins down that information in space and time. So that's why I keep using the word capture. It's because it's like the occurrence of whatever the neurons are doing at that time that we then pick up as gamma activity is kind of like zeroing in on like this some information is happening here that I need to hold on to and like maintain in this very dynamic environment of the brain, so that when the time is right, I can release it or transfer it to another wave in another brain region to then like move it up the chain of command and make sense of it. So they and kind of what's interesting about thinking about all of this is that it kind of when I'm saying it captures that the gamma activity is capturing some information and like knows that it has to hold on to it until the time is right to release it, kind of suggests that in your current neural state, you can also kind of expect what is going to happen next. So what's happening in your brain, like all the waves and how they line up and what brain regions are involved, it's encoding, it's processing not only the current state, but it's also making predictions about or kind of anticipating your future neural state. And so we had all these task stages. We've kind of been narrowing down to like what we've been kind of narrowing down and looking at like very specific relationships between brain regions and like certain kinds of waves and certain kinds of overlaps. And what they find is that there's a certain shift, like the gamma activity will occur in like the late phase of a theta wave. And then if we move from one step to the next of the task, it will shift to another place. But in each step of the task, there are components that we can observe in the waves that represent the current neural state as well as the future neural state. So what's coming next? So let me see. I wrote this down. Let me see if I can find it. So they find that during the context, we're talking a lot about the maintenance phase and then the face presentation phase. During the maintenance stage, there are gamma bursts from the OFC that line up with the late phase of theta waves that are originating in the hippocampus. Then during the face presentation, there are the gamma bursts shift to coincide with the early phase of the theta oscillation, also coming from the hippocampus. So those kinds of late early shifts will become important in understanding what I said earlier about how we can kind of almost concurrently have like a current neural representation as well as a future neural representation kind of at the same time. But really, it's extremely rapid cycling. I believe. I don't know anyway, I'm not going to get into that because it goes into something that I don't fully understand. Okay, so then during with respect to what's happening between the amygdala and the OFC in the context maintenance phase, gamma bursts originating from the amygdala are tied to the late phase of theta waves originating from the OFC, and then during the face presentation phase, those gamma bursts shift to being paired with the early phase of the OFC generated theta waves. I want to like write this down or highlight it in the paper if you're following along. But so the gamma bursts, which the gamma bursts will represent, and their placement on the theta oscillations represent the current and future neural states. The positioning on the theta wave does that. And so, bursts aligned with the early theta phase reflect the current neural state. And then when they shift, and then when they're paired with the late phase, it reflects the future neural state.
Yeah, it's basically it's quite challenging. This paper is quite challenging to understand. But what that is, to summarize, what that means is, so in the theta wave, you can kind of chop the wave up to the rising bit or the falling bit, because that's what waves do. They rise and they fall. They rise and they fall. And roughly speaking, during the rising bit, there you see some gamma bursts. So remember, the theta is the slow one. Gamma is the fast, high-frequency one. During the rising bit of the slow theta wave, if you look at those gamma bursts, you can use that to predict. You can use that to predict what your brain would look like for your current stage of the trial. But if you look at the other side of the wave, like the falling bit of the theta wave, you look at those gamma bursts alone, they seem to match what you're going to do in the next phase of the trial. So that's why they think at any given stage of the trial, your brain is holding different types of information in these gamma packets, in these gamma bursts, and they are separated by which bit of the gamma wave they are riding on. Is it the falling phase or the rising phase? And they kind of verify in using basically a decoding strategy, I guess. That's why I got out of it
Bayesian decoding
from
analysis.
Figure 5.
Yeah.
But they can use this information to predict how the subject would rate the face that they will see in the future. And that's how they kind of, I guess, demonstrate that these packages, these gamma firing is important.
Yeah, so I kind of was diagramming this out for myself. And, I drew one wave, which is one theta wave, which is simultaneously the theta wave generated in the hippocampus, but also the theta wave generated in the orbitofrontal cortex. And I'm looking back to be like, is it really are they synchronized?
Yeah, so that you have to go back to figure 2 to see whether they're synchronized. And also, you also have to go into one of the supplemental figures to see if there's even any theta wave, because the theta wave itself also comes and goes. And you have to keep track of it in each of the 3 brain regions you're looking at. So and then it is a complicated system.
Maybe it is the wave is synchronized, so you could think of it as like a kind of larger or like a more widespread global theta oscillation. But the gamma activity that is overlaid on that global theta oscillation comes from different brain regions. So it's the encoding of the contextual valence is occurring when the gamma activity in the OFC aligns with the late phase of this global theta oscillation that I've created during the context maintenance phase. The carrying over of that information to the stage transition from context maintenance to phase presentation is done when gamma activity in the amygdala is overlaid with this global theta oscillation, but it's the one that's driven by the orbitofrontal cortex. So it kind of makes me think of like quantum entanglement in a way, because the information has to exist in 2 different places simultaneously. Like it's, talking about waves, it is all physics. And so it's probably a spurious an analogy that I'm making, but the information is captured in one place and is not actually physically moved to another place in the brain. Do you kind of see the kind of like mind bend that I'm trying to communicate? Which is that it's not like there's actually like a packet of information that's transferred from like one place to another. I don't think there's much
mystery. If you allow the different areas of the brain to talk to each other, then it doesn't necessarily have to be a mystery. They are just communicating. So the theta wave in, for example, the orbitofrontal cortex might signify some sort of things happening in the orbitofrontal cortex, and that sends axon to the amygdala, causing amygdala cells to fire. And when the amygdala cells fire in bursts, it registers as gamma bursts in the amygdala. And that's why they line up, is because the orbitofrontal cortex is talking to the amygdala, causing it to fire, and then they line up properly. So
I guess that's
in some sense,
sorry. If you look at the diagram of the waves that they have in the like graphical abstract, for example, and you were to overlay that with my own scribblings, it does not show you the actual physical connections between the brain regions, which are the axons. So I guess that's kind of where my like, it's like quantum entanglement where like information has to exist in 2 places at the same time. No, actually, no, it doesn't. It does not have to do that. There is literally a physical passage from one part of the brain to the other. Right. I mean, and that's the axons.
Until you start factoring in ESP, then it gets tricky.
Yeah. So yeah, they were just, so they have this kind of encoding of the contextual valence that's occurring when there's the overlay of gamma activity on a certain phase of this theta oscillation in 2 different brain regions. That information gets carried over and is represented by gamma activity laid on the theta wave in a different spot, different brain regions. As we get into the facial presentation part of the task. And then there is another overlay of gamma burst activity on another phase of the theta oscillation that binds the context to valence information and the face rating together and carries that over to the decision stage. So what they have essentially shown us here is step by step in each of these tasks in their very like reductionist environment of the operating theater. They've broken it down into these steps and said what is happening in the brain at each of these steps to kind of culminate into the decision and the decision of how you feel about that face. And just to take it back to what we were talking about before, like at the very, very beginning, you were doing this all day long. Like we just struggled for like, I don't know, how long has it been? Like an hour and a half, maybe less, to kind of to understand the mechanisms of this. And your brain just does it effortlessly all day long.
I think this is what makes us human compared to, say, a robot, this context-dependent processing.
But could that be encoded? I would argue that yes, probably.
If we know exactly how it works, I'm not sure we're there yet. We have some
recreate
ideas.
It. Yeah.
I think AI
because a robot would react the same way in every situation, not dependent on context at all, right?
The older robots might, but the newer ones with AI, with a huge contextual window that large language models now have, who knows? It's
an interesting question, right?
Do the LLMs now that like you can talk to them, do they and then they respond in kind? Can they currently like interpret
faces?
Like if you say something like that's so
like
great,
tone of voice?
Yeah, or be like that's so great.
Oh, tone of voice. I have no idea.
Yeah, because in that way they could be like,
must be working.
Listen, based on your question and the way you're sounding when you say it, that's going to change the response I give you.
Actually, I have no idea. That's an interesting question, but it's just language model. So right now I'm guessing no. But the production model, so if you use Notebook LM, if you abuse AI to read papers the way I do, you realize that in these fake podcast AI, the speaker have a range of emotion that I think Google spend a lot of care training because that makes them sound more human.
So you listen to these generated podcasts to read a paper?
I can go through like 5 papers a day just
right, but
going about, like when I'm doing shopping, I can listen to a paper.
See, oh my gosh, this is what brought me to TWiN during COVID, wandering around in my backyard being like, I wish there was a way I could consume papers faster because I'm always like cooking or driving the kids or doing bench work and I have to listen. And then I found you guys. It was the next best thing. So,
well, we only do 1 paper a month, so it's not so efficient.
I know.
Exactly. Well, I mean, that's why it potentially has a market.
So, in your opinion, are the summaries accurate?
For Notebook
Factually?
LM, like for this gig.
Yes.
It's okay. It's really useful if you actually don't know much about the topic. And it is a way of getting you a little bit up to speed. I think I've heard someone characterize these AI as, think of it as a friend who knows a little bit about a topic, and you won't just immediately trust the friend, and you would actually fact check that person. Just because he's your friend and you recognize the voice doesn't mean everything they say is correct. So take everything with a grain of salt.
Especially because papers are written in a way as to convince you to. Yeah. So the AI the podcast host says like, yeah, it's just like a really strong, it's just really like strong evidence for blah blah blah, and it's like, well, because that's what the authors think. But
these podcasts are trained to be like super, like they hype things up. So they would, anything you put in, they would say, oh, this is this is groundbreaking research, or this is
really novel,
important stuff. Yeah, exactly. So something.
I asked ChatGPT last week who was the first person to make an infectious DNA of a virus.
Did they get it
So
right?
it said me, and it said for poliovirus, and then it said he took the RNA virus and made a DNA copy, and then he made a transcript of the DNA and put the RNA in cells, and that's not what I did.
You just did the DNA, right? And
I
then
just
it
put the
made
DNA in cells. So I don't know why they got that wrong, because it's all in the paper, right? And then they say it's the first example of an infectious DNA of a virus, an RNA virus. And it's not. Q beta, which is a phage, was done about a year before. So
lies.
But I think they digested all the TWiF transcript, and I think you must have talked about the polio much more than the phage study. So there's a lot more training data. So this is one of the downside of the AI, is that it is training data influence.
But it's always fun to do those queries, because you can, if you're knowledgeable about a field, you can pick up errors, right?
Right, and if you're not knowledgeable, you can get a lot of errors. But still, I think there's some value in that. There's some error, but the big 80% of it is useful information. So,
yeah, but you
don't know which 80%. That's a downside.
Well, that's the thing. Let's say you developed a seminar based on what they told you, and you got up there and said he made a transcript and put the RNA in cells. You're wrong. You're telling them misinformation, right?
Yeah.
So.
Yeah.
So anyway, that's the paper.
That's cool.
I feel like everyone should walk away with like a vague understanding of that this type of research exists, and that it's super cool, but like hard to break into. And I tried to use all my brain cells. But yeah, they're complex questions, because they're just I find that they're difficult to visualize.
I think oscillation is really hard. Oscillation is hard because it does require knowledge about physics, because it's oscillations.
It's physics.
So that's like physics for undergrad physics, which is rough.
This was all a ploy for everyone to go and like spend hours on Wikipedia or something like that, trying to understand, just pull up the Feynman lectures and, book one.
But if anyone is interested, look up imaginary number or like complex numbers. That is a good introduction actually to oscillation, because oscillation uses it a lot, and it's kind of fun to think about people inventing something like the square root of -1. But it doesn't say much about the brain. The brain just kind of piggybacks on these tools. But it suggests to me that maybe it might be worth spending one episode of TWiN to talk about theta precession and like play cells.
Tim.
So this would be like the GPS of the brain. Maybe that would be something worth doing.
I mean, since we've had a couple of, I guess I'm the one who's done them, but a couple of episodes talking about them, and clearly they're of interest to me, and I have stuff to learn. I think that would be cool. But it's all on you, Tim, because
okay.
No, it doesn't all have to be. It doesn't all have to be. On
it might
you.
not be. It might not be next TWiN. I might take a break from this heavy computational stuff, but in the future, I might
let your brain breathe.
I might. It'll be a learning opportunity for me to learn.
Yeah,
more
me
about.
too. Me too. And if you know people who you can fact check with the actual real people.
I'm not sure if I can get Yuri Bajaki to come on the
Probably
podcast.
Not.
And he might come, but he'll unlikely to be talking about introductory basic stuff that would bring us up to speed. He'll probably be talking about like highfalutin, kind of cutting edge research.
Maybe maybe like 6-9 months down the road, when we've reached a level where we can understand what he's talking about.
Sure.
Thank you, Vivian.
You're welcome.
It's TWiN 67. You can find the show notes at microbe.tv/TWiN. If you enjoy these discussions, we'd love to have your support. Microbe.tv/contribute. Tim Chung is at New York University. Thank you, Tim.
Thank you, Vincent. Thank you, Vivian. It was difficult, challenging.
Yeah, I was going to say, don't say a
fun paper,
exciting, fun paper.
But it opens up kind of things to think about. And I still completely failed to remember who examined patient ICHM, so it didn't come back. My hippocampus didn't work. That's annoying.
Vivian Morrison is in New Orleans. Thank you, Vivian.
Thanks, guys, for hobbling along with me on this one. But I think sometimes it's good to challenge ourselves. You know, it's so easy,
of course,
with AI and all of that, to just rely on other people to do the thinking and never explore new ideas, even if we end up thinking about them the wrong way. So, I challenge y'all to challenge yourself and, find this paper, look at the background of the people that they reference, and see what interesting ideas you can come up with.
I think what's interesting too in your life, try and pick out context-dependent reactions if you can, right? Mm-hmm. You have plenty of opportunities all day. It would be interesting to do that.
And maybe frightening if you find that you end up having negative valence ratings on all faces.
Right. I'm Vincent Racaniello. You can find me at MicrobeTV. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.